and you were told that there is a set of laws, set of physical laws, and those laws are named, those laws are, they are known as the Newton's laws. Okay, so Newton laws, we applied the Newton's laws. Okay, if you are throwing a stone, if you are throwing any projectile, the ballistic missiles, everything is governed by the Newton's laws. The mechanics which is proposed by Newton, it is used inside the cars, okay, in the car engines, and all the observable phenomena, they can be very well explained by the Newton laws. Okay, all the civil engineering, it is basically governed by the Newton's laws. सबको ये बात समझ में आई that Newton's laws, they were valid for very broad phenomena that we are observing on the earth, be it construction, be it firing of bullets, or be it anything. Okay, if you are throwing stones, they can be easily described by the Newton's laws. सबको ये बात समझ में आई और इन सब बातों पे आप लोग भी एग्री करोगे. But then we found out that when we tried to describe the electrons, okay, there were some problems in describing the electrons using the Newton's laws. When we applied the Newton's laws to the electrons, we obtained wrong results. We obtained wrong results when we applied the Newton's laws to the electrons or to the subatomic particles or to the atoms which move at, okay, high speed. So scientists, they found out that Newton's laws, they somehow break down at the subatomic level or at the quantum level. At the smallest level, the Newton's laws, they are not valid. Okay, with inspection, the scientists, they come to know that Newton's laws, they are not valid at the subatomic scale, but they are describing everything. Okay, be it the motion of any major particle like a cricket ball, okay, or a bullet fired from a gun or anything, a football. Okay, everything is governed by the Newton's laws, but at the quantum level, we cannot describe the motion of electron using the Newton's laws. So then they found out that we have a new set of rules that is known as the quantum mechanics for the electrons. Okay, and then we applied the laws of quantum mechanics to the electrons, and we developed devices based on the quantum mechanics. So all your phones, okay, your phones, they are operating, they are using semiconductor devices, and there are electrons moving inside these semiconductor devices, and those electrons, they are governed by quantum mechanics. And scientists have utilized, okay, the quantum mechanics to develop all these devices. Okay, be it your monitors, okay, or be it any cell phone, they all are governed by the quantum mechanics. So for the very small scale, we have quantum mechanics. For the normal everyday life, we have the Newtonian mechanics. And then scientists, they also found out that at very high speeds, at the speeds which are comparable to the speed of light, the speeds which are comparable to the speeds of light, then we have new set of laws, which is the spatial relativity of Einstein. And finally, for the greater cosmos, for the universe at a very big scale, we have the general relativity. So everything of them, they comprise the reality. Quantum mechanics is also a reality, spatial relativity is also a reality, and GTR, it is also a reality. And then finally, we studied the standard model of particle physics, which described the smaller particles, and it incorporates quantum mechanics as well. The standard model, it incorporates the quantum mechanical principle as well as the principles of spatial relativity. Okay, so combining the quantum mechanics and the spatial relativity, we have this standard model. So for the complete description of the universe, okay, we have to read all these laws. These are different laws for the different domains. For the very small domain, okay, when we are moving to the quantum world up to the size of the subatomic particles, then we have to use the quantum mechanics, and there is a smooth transition between these theories. Okay, but it is a fact that in our everyday life we observe only those phenomena which are governed by the Newtonian mechanics. तो इसीलिए स्टार्टिंग में आप सब लोग थोड़े अनकंफर्टेबल थे. किसी को कुछ कहना है इसमें? किसी का कोई क्वेश्चन?
Sir, a question, sir.
Yes, sir.
Sir, I request can cloning, sir. Yesterday आपने cloning के process के बारे में बताया था, sir. I request एक बार फिर से बताओ. Just
okay. After the zygote, okay, the zygote is formed by the fusion of the female egg cell and the male sperm cell. Okay, and in the zygote we have the 23 chromosome from the female, and we have 23 chromosomes from the male, 23 chromosomes from the sperm and 23 chromosomes from the female egg. Okay, and this cellular structure and the cellular fluid, it is of the female. This cytoplasm in the zygote, this cytoplasm belongs to the female egg. Very well. So this is the zygote. Zygote has the female cell membrane, the female cytoplasm, the female genetic material, and the male genetic material. Okay. In case of cloning, what we do is we remove these 46 chromosomes. We remove these 46 chromosomes and we insert in this zygote the 46 chromosomes of the person जिसका हम लोगों को क्लोन बनाना है. इन 46 क्रोमोसोम्स को निकाल के 46 क्रोमोसोम्स उस पर्सन के सेल के अंदर डाल दिए जिसका हमको क्लोन बनाना है, एंड इट स्टिल कंटेन्स द साइटोप्लाज्म ऑफ द फीमेल एग. ओके, द सेल मेम्ब्रेन ऑफ द फीमेल एग एंड द साइटोप्लाज्म ऑफ द फीमेल एग. ओनली द जेनेटिक मटेरियल इज रिप्लेस्ड बाय द जेनेटिक मटेरियल ऑफ दैट पर्सन हुज क्लोन वी हैव टू मेक. अर्पित, समझ आया?
Okay, sir, I got it, sir. But sir, इसके जो 46 chromosomes होते हैं, sir, sir, it is of male sperm only, sir. उन्हीं के होते हैं, sir.
These 46 chromosomes, we have taken these 46 chromosomes from the normal body cell. Okay, so the genetic material is entirely same. If we are making the clone of person A, then these 46 chromosomes, okay, we have the genetic material of A.
Okay, sir. Right.
Okay, मतलब 46 chromosomes हमको complete करने हैं. यहाँ पे भी 23 chromosomes female से आए, 23 chromosome male से आए, and there is a diversity. We have, okay, a variety of chromosomes. That is why the offspring is different from his parents. Okay, the offsprings, the children, they are different from their parents because they are having half genetic material from the father and half genetic material from the mother. But if you replace this genetic material and we will replace it with your genetic material, okay, then we will have a new zygote and whose genetic material exactly matches to your genetic material. Is it clear?
Okay, sir. Right. Thank you, sir.
Okay. So the first mammal to be cloned, it was a sheep, and the name of that sheep was Dolly. And the first cat that was cloned, this cat was named as the copycat. Okay, and there is a Bahamas based company Clonate, and this company claimed that they have created the first human clone. So this cloning, it is not legally permitted, and it might even impact the population. It can be used for criminal activities, and the life of the clones, it is short. Now we are moving to the artificial fertility techniques. Artificial fertility techniques. Yes, Nitin, cloning is not sexual. Cloning is asexual. We are doing this whole process inside a laboratory. This whole cloning process, okay, it will be replicated inside a laboratory. Now let us move on to artificial fertility techniques. Artificial. Okay, so a female is producing only one egg in a month, and only if there is a certain kind of hormonal imbalance, only then two eggs should be produced. But in normal cases, only one egg is produced in a month. And in case of males, okay, 500 million sperms, they are contained in 5 ml of the semen, and we generally have 60 to 100 millions of sperms in 1 ml of the semen. This is the normal count, and if the count is less than this, then that person, it will be said that person is suffering from the oligospermia. Okay, so there is term oligo means less. Have you read about the term oligotrophic lake? Oligotrophic lake. You must have read it in environment. Yes, oligo is less, and oligotrophic means a lake which has less mineral content, which has less amount of nutrients. Yes, Arpit, very good. Yes, Palak Mahajan. It is a lake with low primary productivity, and the productivity is low because the nutrients, the supply of nutrients, it is less. Productivity is related to the supply of nutrients. In a similar way, we have this condition which is known as oligospermia, and it means the less quantity of sperms. So if a person is producing 20-30 million of sperms per ml of the semen, then he is suffering from oligospermia, the less sperms. And there is also one condition which is known as aspermia. Aspermia means no sperms. Okay, A plus spermia, and it means no sperms. And then we have azoospermia. You also must have heard the term, okay, zooplanktons. Yes, so here zoo is for the mobile, and azoospermia, it is the immobile sperms. Okay, azoospermia, zoo means mobile and azo means immobile. So azoospermia, it means a person suffering from immobile sperms. And then we have this term which is in vitro fertilization. In vitro fertilization means when the fertilization is achieved outside the human body. Okay, if the fertilization it is achieved in a lab outside the human body, then that is the in vitro fertilization. Okay, the IVF technology or the test tube technology. Test tube baby. Nikhil, what can happen?
Sir, you told us of fertilization outside the human body, so that also can happen, sir.
Yes, in the IVF labs, okay, in the IVF labs, they are doing precisely this. They are doing the fertilization inside the labs in the test tubes. Fertilization is simply the fusion of the sperm and the egg. Okay, so we are achieving this fusion inside a test tube. We are achieving the fusion inside a test tube, so that is why it is the in vitro fertilization. And then the other term is in vivo fertilization. In vivo fertilization means when the fertilization it occurs naturally inside the female body. Okay, the natural fertilization which is happening inside the female body, that is the in vivo fertilization. Okay, and fertilization is the fusion, the formation of zygote by the fusion of the sperm and the egg. Is it clear? We have few terms. Yes, sir.
Sir, a question, sir. Sir, the female, sir, it produces one egg per month, sir. Sir, is it key eggs are produced, they get destroyed, only one egg still remains? Key, it's only one because, sir, if the cell bifurcates, sir, then our egg has 23 chromosomes, sir.
Yes, egg is a single cell and it has 23 chromosomes.
23 chromosomes, sir, and it must have been formed from a cell which must be having 23 pair of chromosomes, sir.
See these egg cells, sex cells, they are produced from the meiosis reaction. Okay, so only one egg cell will be there. Only one egg cell is produced per month by whatever mechanism. There is only one egg cell available for the fusion reaction, for the fertilization. They are produced from the meiosis, and in meiosis the genetic material it reduces to half. Okay, we are left only with the 23 chromosomes. So the mechanism of the body is such that only one egg cell is available for fertilization. Okay, what happens to the other egg cells that are being produced, I do not know. But the females, they produce one egg per month, and the males, they are producing 60 to 100 million sperms per ml of the semen. So what is the difference between sperm and semen? Sperm is the sex cell, and semen is the fluid in which these sperms they remain alive. So semen is necessary for the nutrition of the sperms, okay, so that they remain alive. Now we have certain kinds of banks that are known as the cell banks, and in these banks the sex cells, that is the egg or the sperm, they are preserved in the liquid nitrogen. Okay, they are preserved in the liquid nitrogen at temperatures of minus 190 degrees Celsius, and for this reason, because of the, this is also known as cell banks. They basically produce the cryogenic conditions. Cryo means very cold. Yes, Nikhil, one sperm fertilizes one egg. I told you yesterday that if we have such combinations, like if we have two sperms plus one egg, okay, then it causes instability in the zygote and it dies. And if we have two egg and one sperm. Okay, these combination they are not stable, and the zygote is produced, it dies. So these cell banks, they use the cryopreservation or the cryogenic technology. Cryo means very cold conditions, and in these banks we store the eggs, we store the sperms, and we even store the stem cells. Okay, we will cover stem cells separately, but the cell banks where the eggs, sperms, and stem cells, they are preserved by using the liquid nitrogen. Okay. Now, the artificial fertility techniques, they include that inside a test tube, fertilization is achieved. Okay, the egg, it is fused with the sperm artificially, and in this process, zygote will be formed, and this zygote is then transferred to the womb or the uterus of the female. Okay, inside the womb or the uterus of the female body. Then the zygote, it grows inside the female body. Okay, and rest other process, they are similar to the normal birth. Only the fertilization is achieved inside a lab. ठीक है, सबको क्लियर है, इसमें कोई प्रॉब्लम? Now this IVF, it is also known as the frozen semen technology because, okay, the name is very clear, because we are storing these sperms in the liquid nitrogen at very low temperatures, and this is in vitro fertilization because we are achieving fertilization outside the human body. So this technique IVF, it is used in the conditions if either of the male or female is infertile. If either one of them is infertile, then IVF can be used. Okay, the sperm or the egg, it can be collected from the donor, or the sperm or the egg, it can be collected from any cell bank. Okay, we just need a healthy egg and a healthy sperm, okay, to produce the zygote, and we can source these eggs or sperms either from any donor or from any cell bank. And these cells, so obtained, they are fused artificially in test tubes, and the resulting zygote, it can be introduced inside any woman's body, okay, any woman who wanted to bear a child. Now there is this thing, embryo transfer technology. Okay, first we have discussed what is the normal in vitro fertilization. Okay, and now we are saying there is another embryo transfer technology. So this embryo transfer technology, it is also an example of in vitro fertilization or the test tube technology. The only difference is that in the case of embryo transfer technology, okay, we will allow the zygote to grow into a embryo. We are allowing the zygote to grow into an embryo inside the test tube. So the embryo, it will be generated from the zygote inside the test tube, and embryo is undifferentiated mass of cells. Undifferentiated mass of cells means that we have a single zygote, and from the single zygote, okay, one undifferentiated mass of cells is produced, and this is known as zygote, and all these cells in the embryo, they are alike, and that is why we are saying that they are undifferentiated, because all these cells, they are similar to each other. The differentiation process, it does not begin at the stage of the embryo. Okay, in the embryo stage, the differentiation, it does not occur, and all the cells, they are the same. So again, in this process, the zygote is formed using the IVF, and this zygote is allowed to grow into an embryo inside the test tube, and then embryo is transferred to the mother's uterus. Okay, so the chances of success in case of embryo transfer technology, okay, the chances of conceiving and a successful pregnancy, they are high with the embryo transfer technology. If we will implant the zygote inside the uterus or inside the womb, then the probability of getting success, it is less. But in case of embryo transfer, okay, Minu, I will repeat it. See, in the IVF, what we are doing is, we have the egg and we have the sperm. Okay, one is egg and one is sperm, and in this process, zygote is formed. Okay, in any IVF, this zygote is produced inside the lab, and this zygote, it will be transferred to the womb. Okay, this will be implanted inside the human body. The zygote will be implanted inside the womb. And in case of embryo transfer, what we are doing is, we have the egg, we have the sperm, and then the zygote will be formed inside the test tube. And then this zygote, it will convert into the embryo. Okay, and this embryo will be planted inside the womb. So both of these are IVF because the fertilization is achieved inside the test tube. In embryo transfer technology, we are implanting the embryo inside the womb. This is the difference. Okay, दोनों ही IVF के example हैं. We know embryo is the zygote is the single cell. The zygote is the single cell. The zygote is the result of fertilization, and then this zygote, okay, it will divide, and zygote, it will divide into many cells, and this mass of cell is known as the embryo. Okay, there will be mitosis reaction in the zygote. The zygote will divide. Okay, this mass of cells is known as the embryo, and this is the undifferentiated mass of the cell. All cells are alike in the embryo. Vipul is saying embryo is the fertility seed. Vipul, embryo is. Have you joined late? Embryo is the undifferentiated mass of the cell. The zygote, it will divide. Okay, zygote divide होगा बहुत सारे cells में, and फिर we will have a mass of the cells, और उस mass of cells को हम बोल रहे हैं embryo. और embryo के अंदर all the cells, they are alike. Okay. Yes, the zygote, okay, there will be cell division in the zygote, and obviously it will divide mitotically, and the embryo will be formed. The meiosis cell division. People, you want to ask what is the undifferentiated mass? I am saying that when all the cells are alike, if any cell of the embryo, okay, it is similar to one another. All the cells are identical. So it is the undifferentiated mass of cells. Okay. So we have zygote, and from the zygote, the embryo will be formed. यहाँ पे differentiation process start नहीं हुआ है, and when the differentiation process it will begin, then the different organs will be produced. Okay, different organs with different functionality. That is basically the differentiation. क्योंकि certain genes वो अलग अलग तरीके से अपने आप को express करेंगे हर cells के अंदर. And we will have different organs and the complete human body it will be formed from the embryo. ठीक है, तो एम्ब्रियो के स्टेज में डिफरेंशिएशन स्टार्ट नहीं होता है. द डिफरेंशिएशन इट बिगिंस ओके एट अ लेटर स्टेज जहां पे डिफरेंट ऑर्गन्स डेवलप होते हैं. अब क्लियर है एम्ब्रियो ट्रांसफर टेक्नोलॉजी एंड द आईवीएफ. अगर जाइगोट को ट्रांसफर कर रहे हैं तो हम बोलेंगे इट इज द नॉर्मल आईवीएफ. लेकिन अगर हम एम्ब्रियो को ट्रांसफर कर रहे हैं देन वी विल से दैट वी आर डूइंग द एम्ब्रियो ट्रांसफर टेक्नोलॉजी. और क्या एम्ब्रियो ट्रांसफर टेक्नोलॉजी भी आईवीएफ का एक टाइप है? तो आंसर है यस. द एम्ब्रियो ट्रांसफर टेक्नोलॉजी इट इज आल्सो वन टाइप ऑफ आईवीएफ. और आईवीएफ जब भी फर्टिलाइजेशन एक टेस्ट ट्यूब के अंदर होगा देन दैट विल बी आईवीएफ. आउटसाइड द फीमेल बॉडी जैसे हो रहा है देन वी आर डूइंग द आईवीएफ. क्लियर? अब यहाँ पे क्लियर हो जाएगा. आफ्टर डूइंग द आईवीएफ इफ द रिजल्टिंग जाइगोट ओके or the embryo is introduced inside the womb of another woman, okay, we can after doing the IVF, after doing the fertilization inside a lab, then the resulting zygote, okay, we can transfer the zygote to the womb, or we can wait and we can transfer the embryo to the womb of another woman. Then the woman, that woman is known as the surrogate mother. Okay, this woman is known as the surrogate mother and this process is known as surrogacy. Surrogacy समझ में आ गया? तो जिस फीमेल बॉडी के अंदर we are implanting the embryo, that person is basically the surrogate mother. And there are certain okay issues with the surrogacy and you will study the surrogacy bill, okay, and the fertility artificial fertility techniques bill in the polity. तो जब पॉलिटी में टॉपिक आएगा तो वहां पे आप इसको फ्रॉम द पर्सपेक्टिव ऑफ इंडियन पॉलिटी यू विल कवर द सरोगेसी. एंड व्हाट इज सरोगेसी? हियर आई हैव क्लियर्ड इट. नाउ वी हैव द इंट्रायूट्रीन इंसेमिनेशन और द आर्टिफिशियल इंसेमिनेशन. नाउ इन केस ऑफ द ओलिगोस्पर्मिक पर्सन और एस्पर्मिक पर्सन, वो पर्सन जिसके अंदर स्पर्म काउंट लेस है या जिसके स्पर्म्स जिसमें स्पर्म्स ही नहीं बनते हैं, ओके, देन इन दैट केस वी कैन इंजेक्ट द स्पर्म्स डायरेक्टली इनटू द यूट्रस टू इंक्रीज द चांसेस ऑफ फर्टिलाइजेशन. In order to increase the probability of fertilization, we are injecting these sperms directly into the uterus. Okay, because that will increase the chances of fertilization. Now the isolated sperms, we can also do one thing, because these sperms they are less in the count, we can capture the isolated sperms and we can directly introduce them to the uterus. Okay, the chances of fertilization they will increase. So this process is the artificial insemination, okay, or the intrauterine insemination. Now the scientists they are trying to develop the artificial womb. So this artificial womb, it is a sophisticated incubator like equipment, and this equipment consists of a provision for nutrient supply. Okay, so we have an artificial womb and there is a provision for nutrient supply for the nourishment of embryo. There is a provision for the nutrient supply and we fill this artificial womb with a chemical, okay, with the chemical fluid, and whose chemical composition is similar to the amniotic fluid. Amniotic fluid is basically the fluid which is found inside the womb of a female, okay, womb of a human female. The amniotic fluid is present in which the child grows. So in the artificial womb, we are putting a liquid inside it which is similar in chemical composition to the amniotic fluid. Okay, and there is also a waste disposal mechanism. There is a waste disposal mechanism for this artificial womb. Okay, so we are supplying the nutrients and we are providing the precise environment that is inside the womb by creating this amniotic fluid, and we are making a mechanism to remove the waste generated inside the, okay, inside a living body. So this artificial womb, basically we are trying to mimic the natural womb, okay, by creating a artificial womb. So this is an artificial womb which is in the developmental stage, and this will be used for the humans. Okay, it is still in the developmental phase, and here we have a sheep was developed in the year 2017. Okay, a sheep was developed in an artificial womb. So this packet like thing is an artificial womb, and there are, okay, certain inlets for the nutrients and there are certain outlets, and they are providing the optimum temperature conditions, and a sheep was developed and a healthy sheep was born in the year 2017 by the use of artificial womb. Okay, in case of humans it has not been tested, but in case of animals this was tested in the year 2017, and a sheep was developed outside the, okay, outside the natural womb. Clear? Artificial womb clear? Okay, good. So this was successfully tested on sheep in the year 2017, and this can also solve the problems relating to the surrogacy. Kritika, that you have to Google. You have to Google it. This was in 2017. So this artificial womb, it is still in the developmental phase, and if we can develop the artificial womb, the problems related to surrogacy, they will be solved. Okay, there are certain problems related to surrogacy because we have to search for a willing female person, okay, who is willing to okay, grow the child inside her body. So there are certain problems related to surrogacy if A and B is a couple, A is a male and B is a female, and they have used the IVF technology. Their zygote, it was formed inside a laboratory, and this was inserted inside the third person, the body of the third person, who is C. Okay, so some kind of emotional attachment develops. This person, some kind of emotional attachment it develops with this person. Okay, so there are certain problems with surrogacy, and these issues, they can be resolved if the artificial womb technology it is developed. And it can be used in the conservation biology. Conservation biology means those species which are on the verge of extinction. Okay, we can try to give birth to them inside the laboratories in the artificial form. Okay, and we can also do the surgical interventions, like if the developmental phase, okay, there is some problems related to the growth of the child, then we can do certain kind of surgical interventions and it will be easier for the doctors to intervene in this case. Okay, if any kind of complication is developing inside the body of the child, then because it is visible from outside, then doctors, they can easily do the surgical interventions. Now the artificial wombs, they are of two types. One is implantable and one is the independent. Implantable means which can be put inside the human body. Okay, and independent means which will be put inside a laboratory outside the human body. So this is the independent artificial womb. This is also the independent artificial womb, and we can also, okay, we are also trying to develop the implantable artificial womb. So there are certain people whose uterus is not functional, or doctors have to remove the uterus of such person, then the artificial womb can be used to treat these persons. Now we have one thing which is the womb transplantation surgery, and this womb transplantation surgery is different from the artificial womb. So this is used for the women without a uterus. Those women who do not have a uterus, okay, so this womb transplantation surgery is used for such kind of women, and in this case there is a donor. Okay, the uterus from any donor is taken and it is implanted inside the body of the woman. So here the womb that is transplanted, it is a natural womb. Okay, in the womb transplantation surgery we are using a natural womb, and this natural womb, it comes from a donor. Okay, now there are certain kind of females who were born without a uterus, or there are two fallopian tubes, and if any one of the fallopian tubes is missing, okay, then for such kind of persons the womb transplantation surgery, it can be used. For this womb transplantation surgery, they have been performed with varying success rates. Okay, so this womb transplantation surgery, it is also not, yes Nitin, fallopian tubes, they where the eggs are produced. Okay, the eggs are produced basically in the ovaries, and from the ovaries the fallopian tubes, they carried them to the uterus. You must have seen a diagram like this. Okay, there are two tubes and then we have the uterus. So the fallopian tubes, they are carrying the egg which is produced by the ovaries. Ovaries are the glands. Yes, Palak, very good. So eggs are produced in the ovaries. So womb transplantation surgery, ये भी regularly use में नहीं है, okay, because it is also in the developmental phase, but still it has been performed on the humans with varying rate of success. Womb transplantation surgery को humans को use किया गया है, but the success rates, they differ from person to person. Okay, this is not everyday technology. So in the case of womb transplantation surgery, a healthy uterus is supplied. A healthy uterus is supplied by a donor to the affected individual. तो clear है womb transplantation surgery, artificial womb and the womb transplantation surgery. Okay, very good. Now we are starting with the stem cell technology. Okay. the stem cell technology. So what are these stem cells? I told you earlier that these cell banks, they store the stem cells, they store the egg, and they also store the sperm. Okay. So this technology, it is used to make copies of our organs, or if we try to if we try to create our organs artificially inside the lab, and these stem cells, they are used. Okay, if a part of your organ is destroyed and it can be regenerated, or the scientists, the doctors, they are trying to regenerate a part of the organs using this stem cell technology. Now there is a category of cells which are known as the precursor cells, and precursor cells are those, like if we have the cell type A and if it can convert to the cell type B. Okay, A giving B is an example of the precursor cell. So the stem cells, they are the precursor cells. Okay, remember this thing, A giving B. Okay, A is changing its form to B. So stem cells, they are the precursor cells. So all those cells which can change their form, okay, the A can give B. So these are the precursor cells, and the stem cells, they are precursor cells. Because these stem cells, they can be converted into other type of cells. Okay, the stem cells, because they can be converted into other type of cells. So these are known as the precursor cells. Yes, Kritika, stem cells, they are natural. Now, there is certain category of cells. One is the totipotent precursor cells. So these totipotent precursor cell, okay, one cell, it can convert to any type of cell. Okay, they can change to any type of cells. So those cells are the totipotent precursor cells. Okay, and embryo, the embryo is developing to all kinds of different organs with different functionality. So embryonic cells, they are the totipotent precursor cells. Then we have one thing which is known as the pluripotent precursor cells. Now they can be converted to many types of the cell, but not all the cells. Like we cannot develop the brain cells from the umbilical cord. So umbilical cord, the cord that is attached to the child, okay, during the birth, through which the mother supplies the nutrition to the child. So that part is the umbilical cord. So the umbilical cord, it contains the pluripotent precursor cells. Okay, and those cells, they can get converted into many type of other cells. And the third category is the oligopotent precursor cells. Okay. Third thing is the oligopotent precursor cells, and they can be converted into few cell types. Okay, so the example of the oligopotent precursor cell is bone marrow. टूटीपोटेंट किसी भी कैटेगरी में कन्वर्ट हो सकते हैं. Pluripotent, they can be converted to the many types, and oligopotent, they can be converted to few types. Clear? So stem cells, they are the precursor cells, and all of them are the stem cells. What decides which type they convert to? I think no one has the answer. The embryo is undifferentiated, and at what point the differentiation starts? Okay, at certain point the embryo, okay, and the fetus of all the organism, okay, they are the same. The fetus of all the mammals, they are basically the same, and then through the differentiation, the division occurs. The differences arises because of the differentiation process.
Sir, I mean, in this technology, these cells are introduced in a similar cell kind of environment, and they start producing like using a total cell and embryonic cell will be used and
That is
for
the domain of, okay, that is the domain of strict biotechnology. Only a biotechnologist can give answer to this question. Okay, how they are doing it. But what is the stem cell technology? That I have told you. Because the stem cells, they can be converted into all the type of the cells. So we can, okay, we can exploit this property of the stem cells or the totipotent precursor cells. We can exploit this property of the totipotent cells. Okay, how they are doing it, that we do not know. That is the exclusive domain of the biotechnologists. So like all of you know about the momentum conservation laws. Okay, the momentum is conserved in any process. Yes, Arpit. The preservation of the umbilical cord. Okay, it is an example of the stem cell preservation. So there are certain issues. Okay, we cannot use the totipotent cells. We cannot use them because totipotent cells they are derived from the embryo. They are the embryonic cells, and in the process of acquiring the totipotent cells, the embryo it may be harmed. Okay, and it may even die in the process. If you will try to take a few cell of the, okay, a few cell from the embryo, then it may die in the process. And because the, okay, there are certain legal issues associated with the embryos, because the embryo will gonna develop into a full-fledged human being. So there are certain legal issues and as well as ethical issues associated with it. So this is not legally allowed, okay, to use the embryonic cells. And okay, we can also use the cloned embryo for this process. Okay, direct use of the embryo is not allowed for this stem cell technology because there are certain ethical issues. The embryo may die and it may get harmed. But certain doctors, certain scientists, they also said that we can use the cloned embryo. Okay, we can use the cloned embryo. We will take the egg cell, and inside the egg cell we will, okay, we will put the 46 chromosomes from any other person, and this will be a clone zygote, and we can use the clone zygote. Yes, Meenu. The point is that the cloning is also illegal. Okay, and you, first of all, you cannot produce the clone zygote, and if you have produced the clone zygote, okay, it cannot die. It cannot be allowed to die. The first thing is that it is not legally allowed to create the cloned embryo. Okay, and if it is created, it cannot be allowed to die. So there are both the issues. The creating a clone zygote is also illegal. Okay, and letting it die is also unethical as well as illegal. Now there is something which is known as the induced pluripotent precursor stem cells. Induced pluripotency, as the name suggests, okay, if by some mechanism we can induce this pluripotency property in the stem cells, in any kind of cells, if we can induce the pluripotency property. So we all know that the somatic cells, the normal body cells, they are not pluripotent. Pluripotent, which can convert into all the cell types. Okay, but we can induce the pluripotency in the somatic cells. So the process by which the pluripotency can be induced in the somatic cells, that process is known as de-differentiation. Okay. Sorry, it was my mistake. Pluripotence can be converted into many cell types. Okay, and totipotent, they can be converted into any cell type. There is a difference between pluripotent and totipotent. See, totipotent, they can be converted to all types of cells. We can create the all types of cell from the totipotent cell, and the example of totipotent is embryo. And from the pluripotent, we can generate many of the cell types from the pluripotent. And the example of pluripotent is the umbilical cord. And the third thing is the oligopotent, okay, from which a few cell types can be created. An example of the oligopotent was the bone marrow. The differentiation process was, okay, we have the embryo. We have the embryo, and the embryo, embryo is the cell mass in which all the cells, they are of the same type. Okay, embryo is the cell mass in which all the cells, they are of the same type, and by the process of differentiation, different organs are formed from the embryo, and this process is known as differentiation. Okay, then the scientists, they have realized that, okay, it can be a two-way process. The scientists, they are inspecting that, can it be a two-way process? If by the process of differentiation we are getting different organs from the embryo, then we can also get the embryo type of cells from the organs. So this process is known as de-differentiation. Okay, de-differentiation is opposite of this process. It is opposite of differentiation. If you are getting the different kinds of cells from the embryo, then you should also be able to produce the embryo types of cell from these different kinds of cells. Okay. So the somatic cells, they are not, okay, they are not pluripotent, but we can induce pluripotency in them by the process of de-differentiation. See, we are not using the embryo cells because there are certain ethical issues with it. Okay. If you will use the cells from this embryo, which is going to be a child someday, okay, so there are legal issues with it. We cannot let the zygote or the embryo die. There are ethical issues and there are obviously the legal issues. But if by the process of de-differentiation from the organs, from the somatic cells, if you can create the embryo type of cells, okay, if you can create the totipotent embryo type of cells, then those cells can be used for stem cell technology. वहां पे ethical issues involved नहीं होंगे. Arpit, somatic cells, soma means body. I told in the last class that soma means body, and somatic cells, they are the normal body cells. Apart from the sex cells, okay, the normal body cells, they are the sex cells. तो थोड़ा अटेंटिवनेस आप लोगों की कम हो रही है ना. सोमेटिक सेल्स लास्ट क्लास में मैंने बताया था. और धीरे धीरे कम होगी, बस ट्रैक में रखना। जैसे जैसे सिलेबस बढ़ता जाएगा तो अटेंटिवनेस और ध्यान लगना, ठीक है, एक टायर्डनेस भी होने लगेगी, तो उन चीजों से बचना है। एंड रियलाइजेशन विल हेल्प यू इन दिस प्रोसेस। Because somatic cells, they no see these skin cells, they are somatic cells. Okay, they are the somatic cells. Normal body cells are somatic cells. So you can create my clone by taking, okay, a pinch of my skin. You just need 46 chromosomes to produce my clone. We can induce this property which is known as pluripotency. Pluripotency means the cells which can convert to many types. We can induce the pluripotency in the somatic cell by some process of biotechnology that I do not know. Okay, and that process is known as de-differentiation. The idea behind de-differentiation is that if the embryo can convert to different kinds of organs, different cell types, then we must be able to, okay, we must be able to proceed this reaction in the backward direction. Yes, plug. The simple mitosis reaction produces the somatic cells. The mice skin can regenerate itself by the mitosis cell division. Yes, we are trying to produce pluripotency because for the pluripotency we have only one candidate, that is the embryo, and there are ethical issues in using the embryo. तो इसलिए हमको दूसरे मीन्स ढूंढने पड़ेंगे। अभी सिर्फ एक ही हमारे पास प्रोटीपोटेंट कैंडिडेट है, एंड दैट प्रोटीपोटेंट कैंडिडेट इज एम्ब्रियो। ओके, yes Nitin, they will try to regenerate the organs. Okay, they will try to repair the harmed or the injured organs, and they will try to create even the new types of organs. So now before starting the genetic engineering, okay, there is a break of 5 minutes, 5 to 10 minutes. We will resume the class after 10 minutes. Okay, we are having a break and we will resume the class after 10 minutes, and then we will start the genetic engineering. ठीक है, किसी को कोई प्रॉब्लम है 10 मिनट ब्रेक से? ओके, तो आप लोग भी एक 10 मिनट का ब्रेक लीजिए, एंड देन वी विल स्टार्ट द जेनेटिक इंजीनियरिंग. ओके, ओके फाइन.
Okay.
Hold in. Hold in. Hold in.
Good evening, everyone. Can we resume the session? Is everybody here? Okay, good. Now, Meenu Singh is in the class. Meenu, have you received your copy? Okay, the evaluated one. I think everybody is not here. Okay, so let's wait for 1 more minute.
I got one doubt,
Yes,
sir.
yes, Nikhil.
Sir, actually, in my case, sir, I have optical problems. Sir. Like that is a genetic disorder. According to the when I was born, like my eyes nerves, they are not connected to the brain, sir. Optic nerve.
Okay, Nikhil.
So doctors were saying that using stem cell treatment, that problem can be, that genetic disorder can be solved. Like that they were saying, sir. But people
Okay.
were saying not sure, sir. But then what is the chances, sir? Are there possibilities to solve the problem using stem cell treatment?
Nikhil, the possibilities, okay, the possibilities are always there.
Yes, sir.
Okay, so this is a different domain, and it would be better if the more knowledgeable person than me, okay, who are more knowledgeable in the field, okay, they can comment on it. But yes, by using the stem cells, by using the totipotent and pluripotent cells, the things they can be, okay, we can improve them.
Yes, sir. All kinds
The
of genetic problems they can be solved, sir.
What, Nikhil?
All kinds of genetic problems they can be solved using stem cell.
Yes. See, the genetic problems, they can be solved by gene editing. There is something known as gene editing, and the organs, they can be regenerated using the stem cell. Okay, if your organ is damaged, okay, if your organ is not functioning properly, then that thing can be dealt with this stem cell technology. But if there is a genetic disorder, okay, that genetic disorder, it can be uh, we can rectify it by the gene editing. Okay, we can remove that gene and we can insert a new gene.
Thank you, sir.
Welcome, Nikhil. So I was asking to Meenu if she has received her evaluated copy.
You are not.
Yes, sir, I got it today.
Okay. So before starting this genetic engineering, let us first revise what is this gene. Okay. So gene is any functional sequence of the nucleotide on the DNA. Okay. Any functional sequence of nucleotide. And what was the nucleotide? The nucleotide was nitrogenous base plus sugar, okay, plus phosphate. Yes, nucleoside plus phosphate. So nuclear gene is any functional sequence of nucleotide on the DNA. And not any sequence can be functional. Okay. only certain only certain sequences, okay, they have some meaning and they are the functional sequences. So only 1% of the DNA contains some meaningful sequences. Okay, only 1% of the DNA, it has some meaningful sequences. And genes, they are basically the codes. They are like the computer codes. They are the instructions to the body, for example, to produce a particular type of protein. Okay. People, gene is a functional sequence of nucleotide on the DNA. Like we have the arrangement, like we have this A, T, G, C, C, T. Okay, the nitrogenous bases, they are arranged in a linear fashion. And now say that this sequence, okay, this is a unique sequence, and this unique sequence, it is responsible for producing the insulin. This particular sequence is responsible for producing insulin inside the body. So this particular sequence, this is the gene for insulin. Okay, this is the gene for insulin. See the nucleotides, what are these? These are the nitrogenous bases. And this A, T, G, C, C, these are the nitrogenous bases. Plus the sugar, these strands, they are made up of sugar plus phosphate. Okay, so these are the nucleotides. So nucleotides, basically, these are the sequences of these nitrogenous bases. And any sequence, okay, a sequence which is there for some purpose, it is a functional sequence. And only 1% of the DNA, it contains some meaningful sequences. Okay, only 1% of the DNA, it contains some meaningful sequences. And these genes are basically the codes, they are the instructions for some particular processes. Like we can have a sequence for the production of insulin. Okay, then we will say that this is a particular functional sequence on the nucleotide, and this functional sequence, we will call it a gene for the insulin. No, Subhajit. It is any functional sequence. And any change in this sequence is mutation. Okay. If suppose we have this A, T, G, C, C, and with T, this was the function sequence for the production of insulin. And if we replace the T by G, T is replaced by G, then it will not produce the insulin. This gene is different. So any change, any sudden change in this sequence is mutation. Okay, any sudden change in this sequence is mutation, and mutation is defined as a sudden heritable change. This change will be carried forwarded. Okay, this is a heritable change. It will be carried forwarded to the offspring. So mutation is defined as a sudden heritable change, and basically it is the change in the nucleotide sequence. The nucleotides, they are basically the nitrogenous bases plus sugar plus phosphate. So any change in this sequence is basically the change of the gene, and this is known as mutation. Okay, Palak is saying that according to the Human Genome Project, every gene consists of 3000 bases. Okay. So the sequence is a very, very long sequence, and every gene contains 3000 bases. Very good factual information. Now in the gene editing, what we will do, we will remove a particular sequence in which we are interested. Suppose this particular sequence was responsible for the production of insulin. Okay. This is just an example. This is not the gene for insulin, but I write it as AATGC. So we will manipulate this particular sequence which is responsible for the insulin generation, or that particular gene which is responsible for the insulin generation. Since I am manipulating the genes, so this is the genetic engineering. Okay, now let us again take this example. If due to some mutation in the insulin gene, okay, the production of insulin stops. Due to some kind of mutation, the production of insulin stops, and because the production of insulin it stops, the glucose level it will rise in the blood. Okay, so such a person is known as diabetic. Okay, if the glucose level rises in the blood, then that person is diabetic. And the treatment for diabetes is that we have to provide insulin from outside. We have to inject insulin in his blood from outside. So that is the only treatment. Okay. And obviously the diabetic person, he cannot eat sugar because already he has the enough glucose in his blood. We have to provide insulin from outside to such a person. So how we will treat this person? We will treat him either by using gene cloning, I will explain it, or either by doing the gene therapy. Okay, these are two approaches of genetic engineering. Both are the approaches of genetic engineering to the treatment. Okay, so gene cloning plus gene therapy is genetic engineering. We can say that the gene therapy plus gene cloning is genetic engineering. Disha, you are typing a question. Please go ahead. Its effect to the body. What effect to the body? Of whose effect you are asking? Within it is applicable to all organisms. Yes, the genes, I think genetic material is part of each of the organism, and I think the humans, they are more interested in the human race. I have not yet explained the gene cloning. Okay, I will explain what the gene cloning is. I need a blank slide. Okay, don't worry. So what we will try to do is, suppose there is a person, okay, and this is the cell of a person who is having mutation in his gene, and insulin is not producing inside the body of this person. Okay. Now consider if this is the DNA, the inside circle is the DNA. Okay, this is the DNA material. Don't confuse it with the nucleus. This inside circle, it is the DNA. Now this is the cell of the diabetic person, and this is the DNA of the diabetic person. And there are some issues with the gene sequencing. There are some issues with the genes of this diabetic person. Okay, so what we will do, we will take a normal cell of a normal human, okay, who is normally producing the insulin. So this is a normal person. We have a normal person, and this is the DNA of the normal person. Okay, this is the cell of the normal person, and this is the DNA of the normal person. And suppose on this DNA we have a sequence. There is a particular sequence on this DNA which is responsible for production of insulin. So this portion will be, we will take out this particular portion of the gene which is responsible for producing insulin. So this particular sequence, it was responsible for producing the insulin, and we have, okay, we have taken out this gene. So we have the gene with us. This is the gene sequence which is responsible for production of insulin. ठीक है, सबको समझ में आया? एक नॉर्मल ह्यूमन के सेल में से हमने उस सीक्वेंस को निकाल दिया, इस सीक्वेंस को हमने निकाल दिया. Now there is also one more thing, which is the vector DNA. Now there is also this is not a cell. This new thing that I have drawn, it is the vector DNA. This thing is known as vector DNA, and the biotechnologists, they call it gene vehicle. Okay, this is the gene vehicle. So this is the vector DNA, and this vector DNA is derived from any bacteria, virus. Okay, from these things the vector DNA is derived, because the genetic structure of the bacteria and DNA, it is in the most simplest form. So they use the vector DNA from the bacteria or fungi. So this is the vector DNA. Now this gene, okay, it will be superimposed over this vector DNA. So gene plus gene vehicle, and its product will be this is the new product. By combining this gene sequence responsible for insulin, and this sequence was from the normal person, plus the gene vehicle, we have produced this new DNA. So this is vector DNA plus the insulin sequence. Okay, what is this thing? This is the vector DNA plus the insulin sequence, and we give it a new name, the recombinant DNA. This is the recombinant DNA. ओके, रिकॉम्बिनेंट डीएनए फॉर्म हो गया. इसमें किसी को कोई प्रॉब्लम? See, Minu, we have a healthy person, and from the DNA material of the healthy person, we have taken out the sequence responsible for production of insulin. Suppose if this is the sequence responsible for insulin production, we have taken out this sequence. इसको हम काट के निकाल के ले आए हैं. Then this is the sequence responsible for insulin production. Then there is something known as the gene vehicle. So this thing is the DNA. This is the vector DNA. And we take out this DNA from bacteria and fungi. Okay, so the name of this thing is vector DNA. So we will combine this gene, the insulin gene, with the vector DNA, which is also known as gene vehicle. and its product will be the recombinant DNA. We have superimposed the gene sequence over the DNA of the bacteria, and this is known as recombinant DNA. ठीक है? Very well. Now we will put this DNA, the recombinant DNA, inside the bacterial cell. Now this is the bacterial cell. This is the bacterial cell, and we have put inside the recombinant DNA. Where will be that vector DNA produced? Nitin is asking, where will be that vector DNA produced? See, we are taking out the vector DNA. This is the DNA of the bacteria. Okay, we have taken out the DNA of the bacteria, and biotechnologists, they have chosen bacteria because the genetic material of bacteria and fungi and viruses, they are quite simple. Yes, the insulin gene को हमने वेक्टर डीएनए के बैक्टीरिया के डीएनए के ऊपर सुपर इम्पोज कर दिया. ओके दिशा, तो इंसुलिन जीन हमने नॉर्मल पर्सन से निकाला है, एक हेल्दी पर्सन से इंसुलिन जीन निकाला है. वी हैव सुपर इम्पोज्ड इट ओवर द बैक्टीरियल डीएनए, and this recombinant DNA is basically, it is the insulin plus the vector DNA. Insulin plus vector DNA is the rDNA. Okay, now we have put back this rDNA containing the insulin gene, okay, plus the bacterial DNA. We have put it inside the bacterial cell. इसको हमने वापस से बैक्टीरियल सेल के अंदर डाल दिया. ये जेनेटिक मटेरियल को rDNA को फिर से हमने बैक्टीरियल सेल में डाल दिया. एंड नाउ दिस बैक्टीरियल सेल, ओके, इट विल डिवाइड through its normal process. This bacterial cell, it will multiply. ठीक है, बैक्टीरिया उनकी पॉपुलेशन इंक्रीज होती है थ्रू द सेल डिवीजन, एंड द पॉपुलेशन ऑफ द बैक्टीरिया इट विल इंक्रीज थ्रू सेल डिवीजन, एंड बिकॉज इट इज डिवाइडिंग इनटू सो मेनी कॉपीज, okay, the copy of the insulin gene, it is produced in each of these. The copy of the human insulin gene, it will be produced through replication. ये चीज सबको समझ में आई? As the bacteria will divide, the copy of the insulin gene, it will also be produced through this replication. Okay. Now the industry people, what they do is, they can easily isolate this insulin gene from these bacteria, and from this insulin gene they are producing the chemical they isolate the cloned gene. The biotechnologist or the industrialist, they isolate the cloned gene. This is known as cloned gene, and from the cloned gene they produce the insulin. And the insulin will be produced at the industrial level through this process. Okay, through this process, the insulin will be produced at the industrial level. And this is known as, this production is known as insulin, this insulin production is known as humulin. So the insulin that we are producing in the industries, that is known as humulin, and we are producing humulin through the gene cloning. Okay. Every time the bacteria multiplies, the gene it also multiplies. एक gene से we have so many genes, and from these genes, by isolating these genes, we are producing the insulin from it. Yes, it is by the replication of the rDNA. It is the result of the replication of the rDNA. Yes, Nathan. Yes, we know. Basically, the insulin gene, we superimposed it over the bacterial DNA, and when the bacterial DNA it multiplied, okay, the insulin gene also multiplied with it. So yes, that is the clone gene, we know. The insulin gene, that is the clone gene, because we have created so many copies of the correct gene, okay, through this process. So this thing is the gene cloning. Now, if due to some mutation in the insulin gene, I have explained it, if the production of insulin stops, then the glucose level they will rise and the person will be diabetic. And the treatment is we have to provide insulin to him from the outside. Okay. So we can do this treatment by the gene cloning, or we can achieve it by the gene therapy. In the gene cloning, I have already explained that by using a bacterial DNA, okay, we are replicating the insulin gene, and from that gene we are making the insulin, and that insulin is sold in the market, okay, and which is used to treat the diabetic persons. And then now I will discuss the gene therapy. These are the two approaches of genetic engineering. Now, what is the process? What is the process for gene cloning and gene therapy? I have already explained it. Yes, Subodh. Bacteria, they multiply rapidly. Okay, it will be easier to create the copies of the gene. And the second reason is, Ritika, we are interested only in the human clones. We want that human clones, they should not be produced. That is why we produce the clone of the sheep. Okay, we can produce the clone of the sheep, we can produce the clones of the cats, and we can, okay, we can interbreed lions and tigers. We have produced the liger. We are doing the interbreeding of the tigers and lions, and a liger is produced by that interbreeding. But all these processes, they are banned in the humans. The humans can't do it. And this is the cloning basically of the bacterial DNA. We are replicating the bacterial DNA. There are no ethical issues with it. See, human species, we are a selfish species and we are concerned only about ourselves. Okay. तो यार जब हम sheep का नहीं सोच रहे हैं तो एक बैक्टीरियल ओके बैक्टीरिया का तो क्या ही सोचेंगे. Okay, and even all the vaccines and all the medicines, they go through the animal trials. Now, I was explaining you the gene cloning. So in the first step, what we will do, we will cut the gene of interest from the healthy human cell. We will cut the gene of interest from the healthy human cell, and in our case, we were interested in the insulin gene. And in the second step, we will place this gene over the vector DNA or the gene vehicle. We put this gene of interest over the vector DNA or the gene vehicle, and this combination is known as recombinant DNA or the rDNA. So this is also one of the reason that It is also known as recombinant DNA technology or the rDNA technology. Okay, the name genetic engineering and the rDNA, they mean this one and same thing. So recombinant DNA is formed by superimposing the gene of interest over the gene vehicle. Okay, and the gene vehicle is the DNA material of any bacteria, any virus. Okay, so in the gene cloning, this recombinant DNA is put inside a bacterial cell. In the third stage, what we are doing is we are putting this recombinant DNA inside a bacterial cell, and when the bacteria multiplies, the gene of interest, it also multiplies, and this process is known as gene cloning. Okay, we are creating the same copies of the insulin gene, and this process is known as gene cloning. Then the clone gene is isolated, and this isolated gene is used to produce the required protein from which insulin is obtained, and this all is done at the industrial level. Okay, we are able to achieve this thing at the industrial level, and so many people, they are benefiting from the artificially produced insulin. So we are treating our people by giving them insulin, which is produced inside the industrial process, by the industrial process. Then the second thing is gene therapy. In gene therapy, what will happen? In gene therapy, the first two steps, they are the same. In case of gene therapy, the first two steps are the same. Okay, we will produce a recombinant DNA by merging the gene of interest with the gene vehicle. These two steps are same, and then what we will do, we will insert this recombinant DNA inside the cells of the affected person. That person who needed the insulin, we will inject this DNA inside the cell of that person. Okay, but if you will do it with the grown adult, okay, if you will do it with the grown adult, then you have to provide this copy of the gene to every somatic cell of the body. Okay, if you will give this recombinant DNA, suppose I am diabetic and you are giving this rDNA to me, then what you will have to do, you will have to provide this copy to majority of my somatic cells. You have to insert this copy in all of my cells, and this process is not practical. Supplying the copies of the recombinant DNA to all of my somatic cells, this is not practical. Okay, but we can provide this copy to the sex cells. It will be easier to provide this copy of rDNA to the sex cells, because sex cells, they are one. Okay, there is only one sex cell. If you will provide this copy to the sex cell, then in your offspring, this problem will not be there. This genetic problem, it will not be there in your offspring if this rDNA is provided to the sex cells. Okay. So the recombinant DNA, if this is the recombinant DNA, rDNA, then it will be supplied to the human cell. This is the human cell. This rDNA will be supplied to the human cell. This is gene therapy. If we are doing this, if we are directly supplying it to the human cell, we are doing the gene therapy. Okay. So in case of gene therapy, the recombinant DNA molecule, which is the vector plus the gene of interest. Okay, the vector plus gene of interest, this is transferred to the human cell. ठीक है, डीएनए मॉलिक्यूल को, रिकॉम्बिनेंट डीएनए मॉलिक्यूल को हमने ट्रांसफर कर दिया है ह्यूमन सेल के अंदर, who was suffering from a genetic disorder. Now this gene therapy, it can be done at the two levels. It can be the germline gene therapy, or it can be the somatic cell gene therapy. In case of somatic cell gene therapy, you will have to transfer the gene of interest to all the somatic cells, or to the maximum of the somatic cells. And this is not practically feasible. This is not feasible. So we have to supply, because we have to supply the copy of the correct gene to a huge number of somatic cells. This is not practical. But in case of germline gene therapy, this gene of interest can be transferred to the sex cells or the germline cells. This process is also simple, and is also, it does not have any legal complications. I'm not aware, Nitin, that if we are using this technology in the hair transplantation, but if we are okay, if you are correcting it inside your sex cells, then your offsprings, they will not have the same genetic disorder. There is one difference. Okay, if you are getting bald, आप गंजे हो जा रहे हो because of some gene, okay, due to defect in some gene. But if inside your sex cells we can replace that gene, then in your offsprings that genetic disorder will not be there. No, in case of germline therapy, okay, the affected person, he will not get benefited. And the somatic cell gene therapy, we can do it in certain cases. Like in case of the lungs, it has no legal complications. We are not producing the designer babies. Okay, we are just correcting one gene in your sex cell. Somatic cell gene therapy, it is used in some cases, like if you are having a lung infection, okay, there is some problem inside your lungs because of any particular genetic disorder, then okay, they will provide the correct copy of the gene to your lungs only. Where that gene is required, they will supply the copy of genes to only that organ. So in that case, we can do the somatic cell gene therapy. I will give you example. Now please write the uses of the genetic engineering, gene cloning and gene therapy. The one thing we can write is the production of insulin for diabetes. One thing is insulin production. Okay, thank you, Plug. Plug is saying that germline gene therapy is the insertion of genes at the pre-embryo stage. She is saying that this is done at the pre-embryo stage, that is after the fertilization process and before the cell differentiation. So one use is the insulin production, I have already discussed, and there is one more thing. Have you heard about the growth hormone? We have something known as growth hormone, and this growth hormone is used to treat the dwarfism. Okay, for the production of insulin as well as for the production of growth hormone. Yes, Ashwarya. And for the production of interferons. Okay, interferons, These are the basically they kill viruses. Okay, interferons, they can kill viruses. Kritika, interferons, they are similar to viruses. Sorry, they are similar to antibodies. Okay, antibodies, they kill the virus of specific kind. Like if you have contracted chicken pox, okay, there will be antibodies specific to the chicken pox, and only the chicken pox virus it will be attacked. But in case of interferons, they attack all kinds of viruses. So this is one difference. They kill all the viruses. Generally, they kill all the viruses. Now, by the use of this genetic engineering, we can also treat the hemophilia. Hemophilia. Okay, hemophilia is the deficiency in the clotting of blood. Blood clotting deficiencies, they are related to hemophilia, and this is also genetic disorder, and we can treat it using the genetic engineering. Okay, the vaccines can also be produced from this rDNA. Vaccines can be produced from this rDNA. Now there is one thing, cystic fibrosis. Okay, we have this hemophilia is basically there are something known as the clotting factors. Okay, the blood clot. Blood clot. You all know what a blood clot is. जब भी चोट लग जाती है, okay, the flow of the blood, it stops, okay, after some time because the blood clot is formed. In case of hemophilia, the blood clotting factors, they are absent, and the blood clotting, it does not happen. So that condition is hemophilia. अगर कोई चोट लग गई, then the blood loss, heavy blood loss will occur. I was talking about the cystic fibrosis. Cystic fibrosis basically is a respiratory disease. It is a respiratory disease, and we can cure it by the use of gene therapy. Okay, the correct copy of the gene, the affected individual can inhale the correct copy of the gene. Okay, so the affected organs, they are given the right copy of the genes by the process of inhalation. And the gene vehicle, it can be anything. It can be the bacterial DNA, it can be the yeast, it can be virus. Okay, Now Nitin is also saying that BT cotton. He is saying something about the BT cotton. Yes, Nitin, BT cotton, it is also produced by the genetic engineering through this same process. In fact, all the GM crops, in all the GM crops, we are using the genetic engineering. So BT cotton is one type of genetically modified crop. The BT crops is also a wider category, and within the BT crops we have the BT brinjal, we have the BT cotton. Okay, so BT crops, it is a broader term, and the full form of BT is Bacillus thuringiensis. So please verify this spelling from the Google. So BT is Bacillus thuringiensis, and it is a bacterium. This BT is a bacteria, and this bacteria produces a poison, a kind of poison, and that poison is known as BT toxin. Okay, so we have a soil bacteria by the name of Bacillus thuringiensis, and this bacteria is producing a poison. The name of that poison is BT toxin. Okay, so this bacteria is having a gene which is responsible for the production of BT toxin. So BT toxin, it is producing, this is a protein and this is poisonous, and it is produced from the BT gene. Okay, in BT crops we have BT gene which is responsible for the production of this BT toxin. Now if by using the genetic engineering, what you are doing is you are supplying the BT gene, you have isolated the BT gene from the bacteria, you have isolated the BT gene from the bacteria, and you have supplied it to the seed. Now the seed has the BT gene in it. Okay. Now such a crop, the crop which will originate from this seed, it will be naturally pest resistant because it will produce the BT toxin. Because of the BT toxin, it will be naturally pest resistant. And we have so many kinds of BT crops. We have the BT rice, we have BT maize, we have BT brinjal, okay, and we have the BT cotton. Now the scientific community claims that so where was I? So such a crop, it will be naturally pest resistant. Okay, because this crop, it will be producing the BT toxin. And this scientists, the scientific community, scientists, they claim that the gene, it expresses itself only in the leaves. It expresses itself only in the leaves. Okay, this is the claim of the scientific community. And these crops, they are naturally pest resistant. so the BT crops, they are genetically modified crops, but not all the genetically modified crops are the BT crops. Okay, BT crops, they are only one category of the genetically modified crops. अब यहाँ तक किसी को कोई डाउट तो मुझे बताओ. Okay. So majorly we have covered all of the biotechnology. Okay. And newspapers में ध्यान रखना, things that are frequently in news, they are usually asked in the examination. Okay. And things that are repeating for so many years, okay, they are usually the hot topics for the UPSC. Okay, all these space missions of the NASA that are in news, they are important from the exam perspective. Okay, and if any of the genetically modified crop it appears in the news, it becomes important from the exam perspective. Now yes, Nikhil.
I got one doubt, sir.
Yes, Nikhil, please ask.
So you told that that thing it can kill all the viruses, no, sir. What is that thing, sir?
Oh, that was Nikhil interferon.
So why can't it be used to treat this COVID-19, sir? COVID-19.
That I cannot say. Nikhil, that I cannot say. Basically, interferon is a broader category and which is not virus specific. It can target all the viruses, but interferons, okay, they may not be effective in case of the coronavirus.
And this virus, some people say it is living and some people say it is non-living. So
virus is living inside the human body. Okay, it is on the verge of life and death. If it is inside the human body, it is alive, and if it is outside, it is in the dormant state, then it is dead. Mimi is asking why these BT crops are losing this property of pest resistance. The pests, they are evolving, okay, they are creating their defenses against any changes in the external environment. In environment and ecology, we studied that there is something known as evolution. Okay, and this is the adaptation of the pests to their external environmental factors. This is the adaptation of the pest. And even these doctors, they keep changing the antibiotics to treat us. Because after some times the bacteria, they become resistant to a particular kind of antibiotic because the bacteria, they are rapidly evolving. Okay. So bacteria, they are the simplest form of life and they evolve very rapidly. Okay, and the mutation is necessary for evolution, but mutation is not evolution. Mutation is any sudden change in the gene sequence, and evolution is the acquiring of specific kind of adaptations, specific kind of characters with time. That is evolution. Is tissue culture a part of in biotechnology? Basically, we are producing new products. Okay, if by using the tissue culture we can produce the new product, then that will be part of the biotechnology. The majorly the UPSC, it is interested in this crop kind of things. Okay, and in the healthcare sector. The questions, they will be asked from the current affairs, majorly from the current affairs in the prelims, and in the mains, the question, they will be they will be issue based, wider issue based. So for the mains, the hot topics, they are the AI, blockchain, okay, and the larger universe, because India has given the $12 billion rupees in the Lego India, so the Lego India project. And for the prelims, these kind of topics are important, like the zoonotic diseases that originate from the animals. Okay, if any new technology is in news, okay, anything related to technology is in news, if BS4 and BS6 is in news, then they will be asking questions on it. Things which are in newspaper. And the issue based question, they will be asked in the mains examination. Solar 25. Then in the next class, I will tell you about the Solar 25. Okay. Okay. Because I am not a regular reader of the newspaper, and I am not reading it regularly because मुझे जरूरत नहीं है, तुम लोगों को ज़्यादा जरूरत है, तो इसलिए remain in touch with it. प्रीलिम्स में क्वेश्चंस जो लास्ट 1 ईयर के करंट अफेयर्स है वहीं से ही आएंगे. ओके, कौन सी नई मिसाइल लॉन्च कर दी है, ओके, तो इस टाइप के क्वेश्चन जो फैक्चुअल, मोर the questions that are factual in nature, they are usually asked in the prelims, and understanding based and the issue based questions, they are generally asked in the mains. एंड इससे लास्ट पहली क्लास में स्टैंडर्ड मॉडल को हमने डिस्कस किया. बेसिकली द स्टैंडर्ड मॉडल, इट इज फॉर द सबएटॉमिक पार्टिकल्स. ओके, द स्मॉलर पार्टिकल्स जो फंडामेंटल पार्टिकल्स हैं जिनसे मिलके पूरा यूनिवर्स बना है, वी एक्सप्लेन द फंडामेंटल पार्टिकल्स थ्रू द स्टैंडर्ड मॉडल. ओके, एंड इन द स्मॉलर डोमेन, when we are in the quantum domain, that is when we are discussing the quantum particles, subatomic particles जिनका size बहुत छोटा है, those are the quantum particles. So their behavior is different, and we have to use quantum mechanics to explain them. And for the macroscopic world in which we are living, okay, for this world the Newtonian mechanics, it is sufficient. And for the very high speeds, okay, if we are encountering very high speeds comparable to the speed of light, then we must use the spatial relativity. Okay, and to understand the larger cosmos, to understand the universe, to understand this which are billions of light years away from us, and for this purpose we need GTR, general theory of relativity. So this is the complete description of the universe. These all theories are necessary to understand completely the universe, and we are searching for even the extension of these theories. Okay, there are some questions which have not been answered from these theories. The scientists are even searching for those answers. Okay, for the complete description of the universe, we need all these theories. और gravitational waves, आइंस्टीन की relativity से वहां से predict होती थी, and we have detected them through experiments. And the Higgs boson, that was suggested by the standard model of particle physics, and we discovered it in 2012 inside a laboratory in Switzerland. So this is the broader picture. किसी को कुछ पूछना है लास्ट क्लास से, ब्लैक होल से, इससे रिलेटेड, then please ask. अब बायोटेक्नोलॉजी में जो भी कुछ छोटे मोटे टॉपिक्स होंगे, ओके, आई विल कवर अप, ओके, इन द नेक्स्ट क्लास whatever topics are left, and I have majorly completed the biotechnology. इतना sufficient है आपके एग्जाम के लिए. and things that will be in news, you have to keep track of them. Okay, because UPSC की preparation में किसी भी subject का there is no end to the syllabus. You can always add things. For example, in case of especially the science and technology, we can extend the syllabus as much as we want. But we have to prepare, okay, a certain bit of the syllabus. Yes, Disha. किसी को यहां से तो कुछ नहीं पूछना है ना? Okay. The blockchain and Bitcoin. See, in case of the blockchain, I told you about the distributed database. Okay, a distributed database is a database in which data is stored across the different physical locations. Okay, data को different physical locations पे store करके रख रखा है different computers में, या फिर एक ही location पे there are multiple computers जिसमें हम data को store करके रखते हैं. That is the meaning of distributed database. Very well. Then we talked about the blockchain technology. Blockchain technology, it is a growing list of records, or records, they are basically the digital information. Okay, and these blocks are, they are linked to one another using cryptography. So blockchain के अंदर basically we are using computers, we are using databases to store our data. And blockchain offers us an alternative way to store the data. Okay, and in case of the blockchain, the data, they are compiled in the groups. Okay, and these groups, they are known as blocks. We say we have a block of information. A block of digital information is created, like it can be created for the digital transactions. So for the digital transactions, we have created a block of information. Okay, in this block, the digital information, the information related to transaction, it will be stored. जैसे किसी भी मेमोरी में, किसी भी कंप्यूटर में we are storing things. So this is a digital way of storing information. और in digital blocks को, information blocks को, these are the blocks of the blockchain. These blocks are connected to one another. यहाँ से हमें पता लग रहा है that blockchain is a technology which can be used to store information in a certain way. Okay, and it turns out that the information, once it is stored in the blocks, it cannot be changed. We cannot change it. It is immutable. No one can hack it. So the biggest advantage of the blockchain technology is that it is hack proof. ओके, एक बार इंफॉर्मेशन आपने अपना डेटा ब्लॉक्स के अंदर स्टोर कर दिया है तो उसके बाद वो हैक प्रूफ है. अभी आप क्या करते हैं, लाइक यू ऑल आर यूजिंग स्मार्टफोन्स एंड योर मेजॉरिटी ऑफ योर डेटा, ओके, it is getting saved on the cloud. Okay, if you are saving it on the cloud, basically there are servers on which your data is stored. So your data is digitally stored somewhere. आपका फोन टूट जाता है, then you will take another phone, नया फोन ले आएंगे, and you will be able to retrieve your data from the cloud using certain kind of passwords and other things. Okay, so वो भी एक information storage का तरीका है, and one तरीका जो है, the blockchain offers you an alternative way of storing the information. So what is this blockchain? A blockchain is basically, it is a growing list of records. It is basically digital information which is stored in the form of blocks, and all the blocks, they are linked to one another by the use of cryptography. Okay, And then we explained what is this cryptography. Cryptography, it is basically the one of the technique of secure communication in the presence of third parties. ओके, सो डेटा इम्यूटेबल है, इसका मतलब ये था कि दूसरे लोग उसको चेंज नहीं कर सकते. नो थर्ड पर्सन कैन मॉडिफाई इट. ये तो ना थर्ड पार्टी से सिक्योर है योर डेटा, कोई इसको चेंज नहीं कर सकता. दैट इज द बिगेस्ट एडवांटेज ऑफ द ब्लॉकचेन टेक्नोलॉजी. And in the blockchain, the data, the list of records, it is arranged in a chronological manner, and only at the end of the chain the blocks can be added. तो आसान भाषा में this is the blockchain. Okay. Can you guys give me just 2-3 minutes to complete my tea? Okay. Just 2-3 minutes. I will be back in just 2-3 minutes. Okay. Thank you everyone. I will be back in 2 minutes. So the blocks, is everyone here?
All there.
Yes, sir.
Okay, Nikhil. Some people are offline, like Subhojit, Someranjan, they are offline. Ishwarya is offline. Okay, it's okay. So now we can start. So the blocks, they are attached to one another by way of cryptography, and this cryptography, it is related to constructing new kinds of protocols. Okay, and cryptography is necessary to prevent the third parties from reading your private messages or to manipulate them. आपके डेटा का कोई मैनिपुलेशन भी ना हो, ना ही ओके उसकी आइडेंटिटी मेंटेन रहे. That is the use of cryptography. So in the blockchain, the blocks, they are attached to one another by way of cryptography. Now I also told you that cryptography is different from encryption. The encryption, it was simply the recording of your message. Okay, but in case of cryptography, cryptography is a broader term, and in case of cryptography, the complex mathematical functions and the computer algorithms, they are involved. Complex mathematical functions, theories, algorithm, and computer science programs, they are used in case of cryptography, but in simple encryption is used for the simple coding decoding or using of some kind of mathematical formulas. Then we also discussed that blockchain is a distributed database that is organized as a list of ordered blocks. Okay, blockchain is a distributed database. We know the meaning of distributed database, and the information is organized as a list of ordered blocks, and each of the block is immutable. Okay, information, you cannot change it later on. And this is a distributed append only and time stamped data structure. Blockchain is a distributed, we know the meaning of distributed, when the data is stored across a network of computers. Okay, append only means when the blocks, they can be added only at the end of the chain, and time stamped, each of the block, it has a unique time stamp. Okay, at which time we have added the information inside a block. Block also contains the time stamp when the information was added to the block. Okay, so these blocks, they are basically the blocks of digital information. Okay, these are digital information stored in the form of blocks, and these blocks, they are connected to one another cryptographically. We all know the meaning of cryptography, and all of this data, okay, it is connected to one another and it is secured by the use of cryptography. Blocks are, they are digital information, and each block has certain capacity, like it can be 1 to MB. So this much of digital information can be stored inside a block, and each of the block is assigned a unique cryptographic hash. Okay, a cryptographic hash is like a unique ID given to a particular block. It is a kind of unique identity. Okay, and all of this information is stored over a network of computers. Okay, and there is no central authority in the case of blockchain. No single authority is controlling, okay, all of these computers. No single authority is controlling all of these computers. Okay, there is no central authority. Is it fine? This is the meaning of distributed database, and it is decentralized. Like we have the RBI. RBI is controlling all the banks, so RBI is the supreme authority. But in case of the blockchain, okay, this data is stored on all of the computers. This data is stored on the network, and none of the computer is supreme. Okay, this is a decentralized database. And within each block, the information like the date and time of the transaction, the dollar amount, it is stored in each of the blocks. If the blocks are storing the transaction, the digital transactional information. Okay, किसी को कोई स्पेसिफिक डाउट इसमें? ब्लॉकचेन में बस इतना ही याद रखना है that blockchain is a technology which is used to store data, and in this technology, okay, the information is stored in a way which is immutable. So if there is any specific doubt, please ask me. Okay, otherwise there is no point in repeating everything. Nitin, one block has one hash. Tisha is asking that only parties related to transaction can view it. Basically, once the information is written into the blocks, okay, the information, it is secured cryptographically. Okay, the information is in a different format. Everyone can view it, but not anyone can decode it. It is visible to everyone. Okay, everyone can access it. No one can modify it. Okay, and not everyone can decode it. Yes, Nitin, one block has only one hash. The identity it will be unique. The hash is assigned to a block. Yes, Meenu. One block has the hash of itself, and it also contains the hash of the previous block. And Nitin is asking, encrypted data can be hacked. Yes, if you know how it is encrypted, okay, you can always decrypt it. You can always decrypt it. But it is not possible in the case of the blockchain. Tampering with the hash is not possible. See, hash is created using a special function, and if the data is changed slightly, okay, if the data is changed even minutely, there will be an avalanche effect, and its hash function it will change drastically. It is not possible. It is not possible. The tampering is not possible. You have to basically पूरा का पूरा network ही खरीदना पड़ेगा. Okay, the nodes are there to verify the transactions. The nodes are there to validate the data inside any block. तो अगर पूरे नेटवर्क की स्ट्रेंथ आपके पास है तो ही आप उसको मॉडिफाई कर सकते हैं. Are blockchain used on surface web? What is this surface web? Yes, Disha. For every modification, the hash changes. Suppose here we have written the more on the blockchain. Okay, we are using a cryptographic technique to change it cryptographically, and there is some code for more on the blockchain. Okay, there is a unique code for it. Suppose we have the code 1, 2, 3, 4, 5, 6, and you just you take away this dot or the I. Okay, you have taken away the dot over the I. So this is minor manipulation of the data, and even in that case, the code will change drastically. It will become ABC 1, 2, 4. The code will change drastically. So this thing is known as the avalanche effect. Okay, and if you people are too much interested, then you can always do that certificate course on the blockchain and cryptocurrencies. So the blockchain technology, you have to remember only this much. अगर ये समझ में आ गया है, okay, then it is जितना sufficient है. Yes, the hash will change. If you will modify the information data, anyhow, the hash will change. Yes, Meenu?
Yes, so this hash unique code, we can demonstrate into the diagram that hash of previous block and hash of itself. I can denote it with the symbol hash.
Yes, you can denote it with the symbol hash, but okay,
Okay.
but do not use it in the examination.
Okay.
See, you need not give the examiner the message that you are a software engineer. Okay, you
Okay.
are someone who have done diploma in the blockchain technology. So you have to write answers in a generalistic way. The questions they will be part of general studies question paper. So only this much information is relevant for the general studies part. This is not the exam. Okay, this is not the exam for the coding
Okay, sir.
or
Thank
the
you.
programming. Okay, Meera. Okay, Kritika. See, if any incident happens tomorrow and the surface web, the deep web, the dark web, it is in news frequently, then it will become important from the exam point of view. But for now, it is not very important. And I don't think that even 60 to 70% of the software engineers, they will not be knowing about these things, the surface web, deep web, or the dark web. Because it is decentralized, no one is controlling it. Okay, that is decentralized. If you are the captain of a team and there is one member in your team, okay, who is not under your control, who is not following your commands, will you accept him? You will never accept him. Okay, you will never accept him. So there are trust issues. And since there is no one controlling the blockchain, okay, this is a decentralized technology and government does not like those things which are not in their control. No, if I started the fission reaction, can I stop it? Okay. This blockchain is decentralized, no one is controlling it. Okay, so if we legalize it, then it will be very difficult for us to control it. So if there are any more specific doubts, then please come up with your specific doubts, and whatever we have done in the class, the doubts they should be related to those. Okay, and if you have more inquisitiveness about the technology, we can always enroll in some certificate course on the blockchain, but we are not here to learn the programming. Okay, come up with your specifications. Okay, the bitcoins they are related to blockchain because the bitcoins are using the blockchain technology. So this technology of blockchain, it is used to generate a cryptocurrency. Okay, cryptocurrency basically, suppose there is a digital wallet and there is any digital currency. Okay, and if the digital currency उसको आप spend कर रहे हो, you are spending your digital currency, then even the spending of the digital currency is a data that will be stored in the blocks. Okay, so through the use of blockchain technology, they are ensuring that there will be no double use. The hackers, what they do is they manipulate the electronic wallets. Okay, and by manipulating the electronic wallets, they suppose you are having 50 rupees in your wallet, you have a digital wallet and there are 50 rupees in it, and you have spent the 50 rupees on some product. Okay, and again by manipulating the wallet, because this is a digital technology, you are again spending it. You have spent the 50 rupees on two different products. Because the data in the blockchain cannot be manipulated, so this problem of double spending. The hackers, they were manipulating the digital wallet, and one digital currency, it can be spent twice. The professional hackers, they were doing this. They were manipulating the wallets and the currency, it can be spent twice. But Bitcoin is a digital currency which is using blockchain technology. So with this thing, because blockchain is hack proof and the data is immutable, so with the blockchain, the problem of double spending, it ended. Okay, and precisely how the hackers, they were able to double spend the digital wallet. Okay, this only a blockchain technologist, he can tell it precisely how they were doing it. But by using the blockchain technology, okay, in Bitcoin we are using the blockchain technology. It is hack proof. So the problem of double spending, it is not with the Bitcoin wallet. Yes, Nitin. I also gave one example in the class. Okay, it was used in the in US in the Virginia elections. See, in the West Virginia in USA, in the elections of November 2018, the blockchain technology was used in the voting. Kritika, polar vortex is basically an area of low pressure in the upper atmosphere. This is clear. Point number one, polar vortex is an area of low pressure in the upper atmosphere. Very well. And this low pressure area, this is surrounded by the jet streams. Okay, this is surrounded by the jet streams, and because this is a low pressure area, so from the equator, the winds that are warmer, they cannot come inside this low pressure area. So the cold condition inside this low pressure area, they remain for a longer period of time. Okay, because this encircling jet stream is protecting this low pressure area from the warm air from the tropical latitudes. Okay, then temperature conditions low pressure area के अंदर, they will be sufficient for the formation of polar stratospheric clouds. And polar stratospheric clouds बनेंगे, then from the inactive reservoir of chlorine, the chlorine will be released. और जितनी ज्यादा क्लोरीन होगी उतनी ज्यादा ओजोन डैमेज होगी. ओके, जितनी ज्यादा क्लोरीन होगी, more the chlorine, the more will be the damage to the ozone. So this low pressure area which is encircled by the jet stream, it is the polar vortex. Polar vortex to West Virginia. Need the no, the polar vortex, it is not connected to West Virginia. The polar vortex, it is in the polar regions, in the Antarctica. We have the polar vortex in the Antarctica and in the Arctic Circle. Yes, Vipul, very good. Vipul has given the reaction between the inactive reservoir of chlorine, chlorine nitrate and HCl, producing the nitric acid and the chlorine gas. So this reaction, the ice crystals in the polar stratospheric clouds, they provide a substrate for this reaction. This reaction is, okay, its rate increases in the presence of ice crystals. And the polar stratospheric clouds, they are providing the ice crystals. The rate of this reaction accelerates and more chlorine is produced. तो जितनी ज्यादा ठंडी कंडीशंस होंगी उतनी ज्यादा क्लोरीन प्रोड्यूस होगी एंड क्लोरीन तो ओजोन को डैमेज कर देगी. ओके कृतिका, आपने जब ओजोन डिप्लीशन हुआ था उस टाइम पे क्लासेस नहीं की थी क्या? Yes, Nitin. When the jet streams, they get weaker. Okay, the stronger the thermal contrast between the polar areas and the equatorial areas, the jet streams, they will be stronger. When the contrast is not stronger, the jet stream, they will start to meander. And when the jet streams, they will make this wavy formation, then the polar vortex, it shifts to the south in the northern hemisphere. So we have United States. So the polar vortex, it shifts to the south, and polar vortex, by shifting to the south, it brings the cold air to the US landmass, and because of which we have severe winters in US. Okay, Kritika. See, Kritika, before explaining the polar vortex, I explained the pressure belts on the earth. Okay. I explained that if there is a high pressure on the ground, then it must be supported by a low pressure in the upper part of the atmosphere. Okay. And then I explained the wind system, which is due to three forces: due to the Coriolis force, due to the pressure gradient force, and due to the frictional drag. And then I told you that in case of jet streams, एक friction force वहाँ पे नहीं होती है. So jet streams are also there. And then I went for the polar vortex. So before understanding the polar vortex, we have to understand all these concepts. And now that your geography has started, and I hope that in the next 7-8 lectures, these concepts, they will be cleared there. But you can also watch my earlier videos. I have explained the wind belts of the earth. Okay, this is not rocket science. It can be, you can understand it very easily. The winds, they blow because of the pressure difference, and on the earth we have the pressure difference because of differential heating of the sun. Okay. Suppose earth is tilted like this. Suppose my face is the sun and earth is tilted like this. The top part of this pen is, this is the northern hemisphere, this is the southern hemisphere. So when in this position, the northern hemisphere is, okay, it is closer towards me. And closer in the sense, I can directly see it, and the northern hemisphere, it will receive the direct rays of the sun. And this tilt of the axis, it remains in this way, and earth moves with this tilt around the sun. So don't worry, you will cover this in geography, and these are very simple concepts. Okay, just a 3D visualization is needed. But remember only one thing, that polar vortex is an area of low pressure in the upper part of the atmosphere, and that area of low pressure is protected by jet streams. और जब जेट स्ट्रीम्स उसको एनसर्कल कर रही होंगी तो इक्वेटर से कोई भी वार्म एयर आके पोलर वोर्टेक्स के अंदर वार्म एयर नहीं आ पाएगी, तो वहां पे टेम्परेचर कंडीशंस लो ही रहेंगी और उसकी वजह से पोलर स्ट्रेटोस्फेरिक क्लाउड्स वो फॉर्म होते रहेंगे। ओके, मैंने बताया था ना, PSCs polar stratospheric cloud, they require temperature of -83 degrees Celsius. Any more specification? Guys, mechanism or the process of blockchain tech. See, what kind of mechanism? The blockchain, I have explained you already the blockchain. You want to ask the process of adding a block. See, there are nodes. Nodes basically they are the computers, okay, in a network, and in all of these computers, okay, the digital information, the blockchain is stored. So this blockchain is stored, this digital information, this data is stored in all of these computers. Okay, now suppose a digital transaction has happened, and that digital transaction, okay, it will be recorded inside a block with many other digital transactions. So the content of this block, it will be verified by these nodes, okay, because all of these are strangers, they do not know one another, and neither they do not know who has done the transaction. So they will verify the authenticity of this transaction, okay, they will verify the data from so many things available over the internet, okay, from the data available elsewhere, they will verify the content of this block. Okay, if majority of the network agrees, that content of this block are authentic, this transaction is valid. Okay, this transaction is valid when everyone agrees, okay, by the majority of the network, then this block is added to the blockchain. Okay, so those computers which verify these transactions, the owners of these computers, they are the miners. And the verification process, it requires solving the hash algorithm. Okay, the information is cryptographically secured, and to verify it, the miners, they are solving the hash algorithm. Okay, so they are rewarded for their labor. Okay, the miners are rewarded for their labor. So this process is, the solving the hash is known as the mining. And in order to verify the block, okay, the miners, they will do the mining. Basically, they are solving the cryptographic hash. They will verify the content. Yes, people, absolutely. This process is the mining. The process of adding blocks to the blockchain is mining. And you are doing some kind of labor, okay, you are solving the hash, so you will be rewarded for this process. In case of the Bitcoin blockchain, okay, you will earn Bitcoin for this. And believe me, only, okay, yes, Kritika.
सर मैं ये कह रही थी, जैसे आपने भी बोला कि हम लोग एक ब्लॉक को ऐड करने के लिए मेजॉरिटी ऑफ लोगों को एग्री करना पड़ता है और वो हैशेस सॉल्व कर रहे होते हैं। तो सर एक तरह से देखा जाए तो ये जो लोग हैं जो कि अलाउ कर रहे हैं, दीज आर कंट्रोलिंग द प्रोसेस कि ब्लॉकचेन में क्या ऐड हो रहा है क्या नहीं हो रहा है। तो फिर सर इसे हम लोग डिसेंट्रलाइज सिस्टम ही बोलेंगे।
Centralized system is one where there is one controlling authority. Okay, and the nodes, they are basically strangers to one another. There is a network of computer, there are so many nodes, and okay, my computer, it might be a node, okay, your computer might be a node, so all of these are strangers. ठीक है,
ओके
किसी
सर।
के पास कम या ज्यादा पावर नहीं है, सबके पास सेम ब्लॉकचेन स्टोर है। ओके, किसी के पास एक्स्ट्रा ऑथोरिटी नहीं है।
ओके सर, थैंक यू
दिस इज
सर।
डिसेंट्रलाइजेशन। ओके कृतिक, एंड प्लीज मैं सही बता रहा हूँ कि यहाँ से एक या दो क्वेश्चन से ज्यादा, मेंस के अंदर तो एक क्वेश्चन आएगा, मैक्सिमम दो क्वेश्चन हो सकते हैं, एंड प्रीलिम्स में भी एक या दो क्वेश्चन, इतने ही क्वेश्चन बन सकते हैं। Rest you can use this information in your essays, okay, and UPSC is more interested in the application of blockchain, in the governance, in the in the healthcare, in the property records. So in these things the UPSC is more interested, the application of the technology. How the block is adding, they are not interested in it. Prelims में इतना आ सकता है that this process of adding blocks to the blockchain, what it is called, इतने सिंपल क्वेश्चंस आएंगे। So don't spend too much time. You have the polity to cover, you have geography to cover. Okay, syllabus अंतहीन है UPSC में, so इसीलिए we are giving you the limited material, and this limited material is also too much. ये मत सोचना कि हमने कुछ कम प्रोवाइड किया है। We are providing you so much, but this is limited. लेकिन ये करने में भी आपको बहुत मेहनत की रिक्वायरमेंट है। ओके, जब लास्ट में देखोगे ना कि साइंस एंड टेक का भी इतना सारा मटेरियल इकट्ठा हो गया, अगर इसको रिवाइज नहीं करोगे तो बेकार है। एंड they will not be testing your coding, your programming, how much command you have on the programming. They will not test it. You have the idea that what is the blockchain technology, then how can it be used in the healthcare sector? This can even be an interview question. Okay, so slowly you will understand the demand of the UPSC, and so will understand the nature of work that you are going to do when you will become an IAS. Okay, then you will understand all these things. Is there anything else? If still there is anything else, then please send your specific doubts to me. And ज्यादा फोकस करिए अपने core subjects. The core subjects are the foundation. Okay, जो आपकी base बनेगी, that will be polity, that will be economy, that will be history, and that will be geography. But when it comes to the prelims, you cannot pass prelims without environment, without science, and without economy. So these three things, they are very necessary to pass the prelims examination. Okay, science and technology, it is a part of GS paper 3. Obviously you cannot escape it, because you will fail in the prelims. If you will skip science and tech, you will fail in the prelims. If you will skip environment, you will fail in the prelims. But these pillars, they are very necessary to understand the newspaper very well, okay, and for the broad understanding of all the topics, the things that are going around. Okay, so if I have your permission, can I end the session for now? Okay, thank you very much. So see you in the next session. Bye bye from my side.